Hi, folks. We're going to do a quick sound check. Okay, it sounds like hopefully y'all can hear us online. You can see our presentations, and I wish that y'all could see the bright and shiny faces standing in front of me. But thank y'all for joining today's at RET and REU Symposium. We're excited to welcome our 2026 cohorts of students and teachers who have been working with us for the past 5 to 10 weeks. First in our agenda, we'll hear from our RET teacher participants. They'll share what they've learned from the lab and how they've incorporated that into lesson plans. We'll take a 15-minute break and then hear from our REU students about some of the research that they've been doing with grad students and faculty mentors from across PreMiEr's campuses. Without further ado, I'm going to hand it over to Dr. Megan Hill.
Thank you so much for coming. It has been such a wonderful experience working with the RETs this summer. It's been my first year leading this program, and I think I probably just got the best possible cohort I could have, and I'm very grateful for them and all of the hard work that they've put in this summer. I also want to thank all the mentors and the PIs that were there and dedicated their time and supported the RETs this summer. And with that, we'll kick off with our first teacher, Marvin Smith.
Thank you. All right. Good morning, everybody. Yeah, good morning, everybody. I am jump starting the presentation off. I'm the first RET participant. My name is Marvin Smith. My project, I guess you would call it, is called Measuring the Unmeasurable, the secret tool scientists use to study cells and microbes. Again, my name is Marvin Smith. I teach at Next Generation Academy in Greensboro, North Carolina, where I currently teach 5th grade science, and I love it. This will be my 8th year teaching this academic school year. Over the summer, the past 5 weeks, I've had the opportunity to work with Dr. Misty Thomas at North Carolina A&T State University, where I'm a proud alum, actually class of 2016. So it was great to go back to my alma mater and spend some time learning some new things to help me become a better educator. What we worked with in the lab was bacteria and metal resistance. Our goal was to determine if E. coli resistant to silver nanowires are resistant to silver nitrate. And we used the method of minimum inhibitory concentration. So coming from 5th grade science, we do not do minimum inhibitory concentrations, a lot of those. So it was awesome. Those are real deal pictures of me in the lab working, making the E. coli, using the different tools within the lab. Very great experience. With that experience, I had the task of bringing it on down to 5th grade science. So with that, I have created a lesson. This lesson is to look at something that I took a part of, which was the accuracy of using the tool of a micropipette, which I come to have loved using in my time in the lab. The purpose of the lesson is to kind of close out a physical science unit that I teach in 5th grade science as we enter into the life science unit of 5th grade science, looking at the cellular organization of cells. All right, and my lesson is designed for 20 to 22 students, and that is between 70-minute blocks. I teach 3 blocks of students. The objectives, pretty much with the objectives, we define and explain the difference between precision and accuracy in scientific measurement, which I thought would help out with 5th grade math as well, just trying to tie it all in together. Using P20 and P200 micropipettes correctly, bringing in some different scientific tools that 5th grade scientists can use. Using the claim evidence reasoning framework, which is a highly recommended framework for students to engage in learning how to think and ask questions, and providing them with some real world applications and careers that they can use with the project. With the content standards, we use the 5th grade science standards from Next Generation Science Standards. That's what we use in North Carolina. The physical science units, pretty much for physical science in 5th grade is looking at physical and chemical changes. What are the differences? And again, going into all living things, knowing that the cell is the smallest basic unit of life. And on. These are just some examples of those actual standards that I teach. Carry out investigations to compare the weight of objects before and after interaction is the standard. And myself with my students, we go through and actually define each term within those standards. That's how we break it down. Those are the other standards that we'll be doing. And this is the standard that it will be introducing, using models to recognize the organizational structure of humans as multicellular organisms. We just recently changed standards. So this lesson will not only be on current grade level, but it will also be an extension to 6th grade science, starting to look at unicellular organisms. All right, so here we are into the actual lesson. Day 1, engage and explore. We use the 5E model. That's engage, elaborate. So engage and explore is happening on day 1. Why precision matters, comparing two scientists scenarios. So looking at the difference between pouring versus using a micropipette. And the discussion between students, why does precision matter? When should a scientist pour? And when should a scientist micropipette? That would be a discussion. Typically in the classroom, students would turn and talk amongst themselves, or even do a sticky note response. The exploration part of it all would be station rotation, where they actually use micropipettes, learning how to handle and calibrate those. That will start with a demonstration. The students would then practice measuring 20, 50, and 100 microliters of water onto white paper, and that'll be rotated between two groups. As you can see on the side, I have done the experiment, not in the fanciest way, but just as a way to experiment. And as you can see, I made a mistake and I actually did something wrong at the end, but I wanted my students to see that I made a mistake. That also helps them out too. That day, you will also have students recording their observations. What did they find? What did they see? And to also discuss, how do you know if a micropipette is actually accurate? Cleanup preparation for the next day. Day 2 is an exploration day. Day 2 is more so of a, exploration day. Color mixing challenge day is station rotations. They will actually be looking at the physical and chemical changes, looking at color changes, mixing red and yellow to, of course, make orange, blue and yellow to make green, and red and blue to make purple. With those groups, they will have to mix the precise amounts, 10 microliters of each. So we're looking at making sure they're accurate, as well as looking at the color changes. They would also have to record their data. And then the explanation piece would be, why do we use the micropipettes instead of pouring? And finally, on day 3 is kind of like the escape room challenge, or the finale challenge of Save the Sample. The students will have a scenario where they have a tiny sample of important biological material, and you have to transfer it accurately into multiple tubes without losing or wasting it. The challenge will be to maintain accuracy across all transfers without leaving or losing a drop. The stations will rotate. And then they have the CER, activity, claim, evidence, reasoning, where they receive a claim. Scientists should use micropipettes instead of pouring when measuring small volumes. If you notice, it's a pattern. The more you repeat it, the more it sticks with 5th grade. And that also helps you with your standards mastery. Along with an exit ticket, why would a scientist use a micropipette instead of pouring? All right. The next portion is just differentiation within elementary school and K-12. We always have to differentiate our learning for our different learners that we have in the classroom. So starting off with our advanced or gifted learners, they would have some more so extended practice, having an activity called Pipette the Rainbow, where they actually have to micropipette the different colors into the rainbow. Having to complete the Save the Sample challenge utilizing different multiple micropipettes instead of just one. And then also independent lab exchange work, which is an awesome resource that I received from Dr. Misty Thomas's lab. Grade level proficient learners, which is where you exactly kind of want your learners to be. Just making sure that those clear demonstrations are provided, guided discussions amongst themselves or their peers, and small group rotations within the teacher and their peers as well. We also have emerging or below grade level learners. That's just making sure that they have that vocabulary piece down, graphics, sentence starters, and teacher support alongside. All right, and finally, I have come to my reflection. I am super grateful to have been afforded this opportunity to work with PreMiEr this summer. With elementary, we always have math and reading PDs, but not science. So I was very grateful to have an opportunity to grow with my cohort and with my lab at A&T. This was a very humbling experience because I had to humble and be a student versus being the teacher, and being in that place where I had to ask questions where I did not feel comfortable, and just, being like Nike and actually doing it. And I think that's something that we push our students to do, and just pushing that confidence to learn and grow. And I did want to say thank you. This program has left a stamp on my teacher heart and filling my educator cup, especially with the nice PreMiEr bottle that we received. And I would also like to share this video. This is a small snippet of a video that I hope to receive with my students. This is me actually micropipetting for the first time into gel electrophoresis. And I am very focused, very focused, very focused. And soon the aha moment happens. So that's my response and my reaction, and I'm hoping that my students will give me as well. And that is the end of my presentation. Um, Thank you. Yes.
It's like the precision and accuracy with reproducibility. And I know you mentioned the communication style, but the communication practices are also involved in it.
Yes, so it's also a building process. And for me, implementing this is my first kind of round of teaching fifth grade science. I've just gotten my feet really, really well into it. So implementing those hands-on activities step by step, until I'm comfortable with it. That's something that was pushed teacher clarity. So whenever I feel clear and can expand, that's what I plan on doing with the students. But I am excited about it though. Thank you. Wonderful. Thank you.
Next up, we are going to have Deanna McLemore.
Yes.
Excited.
Good morning. Good morning, everybody. All right, so hopefully I don't have any technical difficulties up here. All right. All right, again, I'm Deanna McLemore. I am also an alum of North Carolina A&T. I'm way older than Marvin, so I am not gonna say the year I graduated, but I am also an alumni of A&T. And I currently teach at Ignite Academy, where I will be teaching physical science, biology, and forensics for the first semester. So fingers crossed on that. Okay, so this summer I had the pleasure of working between two different labs. I was in the Brown and Turpin lab, where I was able to work on projects that were led by Megan and Clara. Thank you so much, Megan and Clara. Okay, so my project in the Brown lab was using the aerosolization of bacillus bacteria to inhibit the growth of mold. So essentially, the bacillus bacteria would be emitted through a humidifier with the hopes of inhibiting the growth of mold. This specific research was very interesting for me because for the past 9 years, I've taught physical science. So some of the microbiology techniques were either a refresher to me or just brand new. So thank you all to the team who helped me and walked me through some of the different techniques that I had just not seen in a while. So for this particular lab, I helped with sample collection, spread plating, and down here, this is a picture of me and Niobe doing some cereal dilution for culture concentrations. And thank you, Niobe, for helping break down some of the concepts to me. She was my right hand with going through some of the topics there. Okay, so for the Turpin lab, the project that they were working on was using a dynamic flux chamber with VOC sensors to detect the presence of mold. So the goal with this is to use robot technology with similar sensors to detect mold VOCs before they're actually visible. Here you'll see me inoculating different building materials. We did drywall with paint, as well as wood to place in the chamber with hopes of some of the VOCs being detected. All right, clicker, here we go. Okay, so on top of doing some actual like lab research, me and Megan did some in the field research here on campus, and we went to Wilson Hall. I see some of y'all's faces already looking at the mold. So we wanted to take a look at some of the environmental conditions of mold growth. And as you see right here on some of the cooling pipes, like all of this black stuff right here is actually mold, as well as you can see the mold that's visible on the vents here. And the main cause for the mold here is that they don't have any central AC in this building, as well as the heating and cooling systems aren't well controlled. So currently the remediation that's being done for this building is they did have small AC strips in the building, as well as a couple of dehumidifiers around, but that was mainly the remediation that was being done in Wilson Hall. So it was nice to do some in the field research just to see what the environmental conditions of mold actually look like. Okay, so with both projects being centered around either inhibiting the growth of mold or early detection of mold, it just kind of made sense to see what the actual mold remediation process looked like. So I did do some background research on commercial mold remediation, and I got the chance to speak with Mr. Ed Robinson. He's a professional mold remediation specialist. And he talked through the actual protocol of what mold remediation looked like. And the key takeaways that I got from my conversation with him was that it's very important to tackle the underlying moisture issue, because if that's not tackled, no matter how much remediation that you do, the mold issue most likely will still return. He said, regardless of the species of mold that they're remediating, the protocol is usually the same. The complexity usually just comes from the location of the mold. Okay, so to transfer my research into my lesson plan, I do plan to incorporate it into my ecology unit. So the two standards that it relates to is students will have to explain how organisms interact with both living and non-living components of the ecosystem, as well as explain how environmental conditions affect the growth, survival, and distribution of organisms. So my lesson plan, I broke it down into 3 parts, and I don't think I mentioned the school that I work at, I recently transferred to, is a virtual school. So I had to gear my lesson plan to fit that environment. It can still be used in an in-class environment, but it's mainly geared towards my virtual school. So for part 1 is an introduction where students will learn the basic characteristics of fungi. Then they'll move to a virtual home tour where they'll learn the environmental factors required for mold growth. And then afterwards, they will do an in-home mold investigation in their own homes. So they will have to predict the locations where mold growth is likely to occur based on environmental conditions, and then they'll have to justify their predictions based off the prior knowledge from the virtual home tour. Okay, so instead of clicking through my entire PowerPoint presentation with the virtual tour, I did add in screenshots of what it would look like. So for the introduction here, that's going to be more delivered in a discussion/lecture format. Most high school students, they already know what mold is, just not really from a scientific aspect. So the discussion will really be led by seeing what they already know and then breaking it down into the science. So where does mold come from? What are the conditions needed for mold growth? What are some of the common causes of mold growth in the home, as well as some of the negative health effects that can occur if mold is not remediated? So the second step of my lesson plan is actually the virtual home tour. So I was able to use a website to create a virtual home tour. This is screenshots of what it would look like. So the the Mode Explorer map right here, students will be able to click on the different rooms here at the bottom, which will then open what you see here. So right here is a visualization of an attic, and it'll be different icons throughout the attic. So once they click on the different icons, they'll receive different pop-up boxes. The pop-up boxes will then let them know the environmental factors as well as the biological explanation of why mold is growing in that specific area. So this part of the lesson will mainly be used as guided notes. They will have to do this checklist over here where they are, again, listing the environmental factors as well as the biological explanation. And lastly, I think what will be the fun part for my students is the in-home mode investigation. Yes, they do have to receive permission from their parents. There is a safety hazard on the beginning part of the lesson plan for them not to touch mold, not to go scraping around trying to remediate their own homes of mold. But essentially, they're going to go around and look for different locations where mold is prone to grow. They'll have to mark down the conditions that were observed, whether it's a low, medium, or high risk of mold, as well as the biological evidence that they saw for the reason why they picked that location. And then the fun part, they'll be able to upload photos of the locations that they decided to pick. There'll also be a reflection questionnaire at the bottom for them to summarize their learning. And that is my lesson plan. So I wanted to give my thank yous out to everybody that I work with in the Brown and Turpin Lab, Megan, M, Niobe, and Will, Dr. Megan Hill, and the rest of my RET cohorts. I think I was. I'm so sorry. Definitely forgot about that part. Okay, yes, Clara, I'm so sorry.
Can you have a question for the
Oh, sure. Okay. So the question is, am I going to offer advice to my students if they are to find mold in their homes? And my biggest advice will be to tell your parents. That will be the number one advice, especially don't touch it, because I don't need those repercussions like, oh, my teacher told us to remediate our own mold in our house. Like, I did not say that. It is in bold on the lesson plan, do not touch mold. So to tell their parents, that will be the number one thing that they should do. Yes.
Student might be embarrassed if they do find mold in their house and not report it to parents.
So the question is, do I have concerns that students will be embarrassed if they find molds in their home? To be honest, I'm the type of teacher, I do like to set the environment, like regardless of what it is, there's no embarrassment for anything. So whatever lesson that I teach, I kind of preface it with that tone. And if they do feel the need to, if they do feel embarrassed, I will advise them to speak with me one-on-one, and we could possibly find some other assignment alternatives. But again, there's no room for embarrassment in my classroom, so. Okay, now I can go sit down.
All right, next up we have Teresa K, or Miss K, as she's known by her students.
Okay. Hoping I'm gonna have this. Yes.
All right, my name is Teresa Kagodo. I teach in the Dumpston County High School, and I was privileged to be part of the PreMiEr. I was working at Duke with my mentor, Chrissy Massimo at JASED Lab. Okay. So the background of what I was dealing with is about phages, and my mentor is trying to look for remediation on the pit traps found in the hospitals. They grow a lot of bacteria under there, and disinfectants and also antibiotics that we use are not able to get the bacteria because they build a layer of biofilms. And this exposes patients to different bacteria infections. So the bacteria phages are different viruses that are very specific. They infect as certain bacteria, and we can use these bacteria phages to control the biofilms. So the goal of this was to isolate, characterize the bacteria phages from the wastewater. The phages are kind of gold, but they are found in the dirtiest place that you may ever think of. And one of it being wastewater, soil, etc. So the study area and materials that we used, we got primary sludge and also activated sludge from the Durham water reclamation facility. We used two main bacteria, Klebsiella and Pseudomonas, and different lab materials. So how are we to deal with these wastewater to finally get the gold, that is the phages? So we have the sample, we process the sample. It's kind of grossy, but that's the process in the getting the gold. We filtered it, prepared the sample, and then we inoculated it for 24 hours, filtered the lysate, did the spot assay, got positive phages, and we purified the plates. It's interesting that you can do all this process and finally not find phages. So we were glad to find the phages. So which are the methods that we used? We did the spot assay. So to notice if there's a phage, it clears all the bacteria. It's like it has killed the bacteria. So if you see these small dots over here, it means there are phages in there that have killed the bacteria. That's why there's a cleared zone. So the next thing we did, we had a plaque assay to purify the phages, and that was followed by plaque purification. So you got to notice where there's a clearing zone. You pick that plaque using a tip of a pipette, and then you elute it, you serial dilute, you infect it with the bacteria, and then you plate. Finally, you count the phages. We also did the spot titer, where we determined different PFUs. From the very last serial diluted number 7, I've calculated the PFUs here. In 0, 1, all the way to 5, around there, it's so hard to count because there are many. So the ones that are less are the ones we use to count. So how am I going to implement this in my classroom? I will use a bacteria that I'm gonna buy called basic bacteriophage culture set A. Reason being, students are not allowed to swab. I wish they are. It would be super interesting to hunt the phages, but now due to restrictions, I'm gonna buy the bacteria and the phages. So we're gonna use E. coli, which is not, or which does not infect human beings, which will be safe for my students. So we're gonna do phage isolation, plaque assay, phage therapy as a biotechnology application. They're gonna learn how specific phages are. They'll do a lot of micropipetting, aseptic technique, LB media preparation. They'll do bacteria growth and a lot of culturing. So which are the investigation we're gonna have in this process? So as I mentioned in the previous slide, they're gonna prepare media. They will know that this is where the bacteria grow. They'll do the enrichments, the plaque assay. They're gonna pick the individual plaques, they propagate them, and they collect and do the spot assay analysis. So how does this align to the NC standards? So I teach biology and also biotechnology. I'm planning to use this in the 2 classes. So in biology, we have biotechnology applications, so where the students need to learn how this is applied. So we're gonna use the phages in application of medicine. Bacteria nowadays are very resistant to the medicine being used. So they're gonna use the phages to discover how medicine is used, where bacteria are resistant to medicine. We also have biology 2.1, where they learn about cell growth and reproduction, and also natural selection. These ones, the phages are kind of the best way to teach about how antibiotic resistance, creating alternative ways of using the phage therapy in different treatments. So my acknowledge goes to the RET team, the team at Duke that I worked with, the one who gave the samples to use in the process, my mentor, Chrissy, and my school that will allow me to implement this. Thank you, and I'm finally a phage girl, and it was nice to meet you. Okay, questions? Yeah, the students are in grade 9 to 12, so they are between 14 years to 18. Some are even 20 years. Yeah, I'm planning to buy a fridge to store them. Currently, we don't have, but the RET team supports us with some funding to buy such equipments.
All right, next up, we have Philip Boyne. Remember when people ask questions at the end?
Yeah, thank you so much. Hello, folks, welcome. So my name is Philip Boyne. It's nice to meet you all. I'm working on an environmental sensor. So that's my plan. And I worked in the Gunch lab this summer. So a little bit about my background. I've taught for 17 years at St. David's. You can see on the top right, that's an aerial drone shot of our school. It's near North Hills in Raleigh, if you're familiar with Raleigh. I've taught physics there for 17 years, and I've also taught engineering for about 10. Every year I do a big lab, which is just my name for a big open-ended project. Students have 6 class periods to do whatever they want. They ask me lots of questions. Can I do this? Can I do this? I say, do whatever you want. They have to write a proposal. So I formalize it a little bit about a science project of some kind. And then they have 6 days in class, and then they present. To give you a flavor of some of the big labs we've seen before, these are a selection of 5 pictures I took from this March. I think I had 22 groups this year do big labs, so these are just a few from this year. On the left is the claw machine. They use syringes. You can see a little bit of the syringe to make a claw move left, right, up, down, and in and out. Then we have a hot air balloon out of a little torch. They did not light they almost lit the bush on fire, but they did not. They're very, very safe. We had a hydraulic press also with syringes, a water gun that used, there were two AP chemistry students. They talked about Boyle's law and combined that with the Bernoulli principle. And they predicted how far it would shoot and the speed it would shoot a stream of water. And then that last group there, they built a carnival ride out of an Arduino and a stepper motor. I also do labs in engineering or projects in engineering lab. So then about my background here this summer, I worked in a microbiology lab with Dr. David Singleton and Mara Chen-Goldberg, and it was awesome. I had a great time, though the direct application of what we did doesn't apply very well to physics. I didn't want to come from this experience and then say, okay, in the middle of our energy and momentum unit, let's use micropipettes and let's pipette different fluids. It wouldn't quite work. So, but all the other stuff we did, I think, is very relevant. For example, we practiced the scientific process. We practiced measurement. And I was really inspired by our trip to the PreMiEr home at Duke. There's a picture of that there, where they were conducting measurements of the environment and seeing how that affects the microbiome in a home environment. I thought that's so cool. That's something we can definitely do. High school students would be very interested in this. So I was really charged by this idea. And I like things that are DIY or do it yourself. So these are materials that I wanted to use, and I brought them here to show you if you can see on the camera or in front of you here. This is an Arduino Maker. This is the microcontroller we're using. It's $40, and you can keep using it over and over again. The Arduino is, it basically interfaces with the computer to do tasks like turning on lights or making a motor move or all kinds of different things you program it. And then the other chip, this is the sensor we use. So I first looked up the sensors that they used at the PreMiEr home at Duke. They were a little pricey, like $150, $200. And probably pretty accurate. I thought high school students don't need that level of accuracy. Let's try to find something a little cheaper. So we're going to use these. These are the Nikola Sense. They came out in 2024, so they're pretty recent. And it measures a lot of different things. You can see there from temperature, humidity, volatile organic compounds, or they call them TVOCs for total volatile organic compounds, carbon dioxide, nitrogen dioxide, ozone, and ethanol. And that's just a schematic of the chip there. So the basic idea of the project is students would use hardware, which they would connect these together. And I'll show that in a separate picture. It's extremely simple. And software, which is the code. The code is also pretty simple. I'm thinking of this as a gateway, a first introduction project. Out of curiosity, do any of y'all, hands up if you're at all familiar with Arduinos, have you used them, a handful of you? Most of you haven't, and that's probably more so the case in my high school classroom, and that's okay. I don't expect students, they'll come in and be scared, and I say, don't worry, we'll learn it together. It's really not that difficult. Honestly, all the code is online, so you can just download the code there and then make edits if you need to. I wanna teach the students about experimental design. For example, they might want to know about CO2 levels, how they change in the day. And they might ask, what happens to CO2 levels in a day? That's a valid question, but it's not a very scientific question. So I thought helping them frame their questions to make them more testable is part of the scientific process. So for example, how do CO2 levels vary in an average classroom afternoon as occupancy changes? And you would expect to see the levels of CO2 rise as the classroom fills and then decay as students leave the classroom. That's something you can directly measure with these chips. Also in this project, you can do data processing, thinking about what you do with your data. How do you display your data? Should you filter your data? And they'll also culminate in a presentation. This is some pictures of me actually using the device. On the top left, I took the Nikola Sense. I held it in my hand and I started breathing. You can probably see in the graph where I started breathing. I cupped it in my hands and did that. You can see the temperature on the left axis jumps up a few degrees, and the humidity jumps up quite a bit as well. So I was like, okay, this sensor seems to work pretty well. The bottom left is a picture of it being used. It's really simple. I just used a cable it came with, and I bought a USB to micro cable. That's all the technology I used. There's a quarter for comparison. On the top right is the indoor air quality test. You can see it measures some indoor air quality parameters there. Such as like the CO2, the total volatile organic compounds, as well as ethanol. And I don't think these are super accurate. Some of these are based on changes from the baseline. It's important to communicate because high school students might think these are God-given values that are completely accurate, 100%. That's not the case here, probably. And then the outdoor air quality sensor as well on the bottom right, you can see it measures ozone and nitrogen dioxide there. So a little bit of the takeaways. Really what I'm doing is I'm secretly teaching students experimental design, scientific thinking, data analysis, and coding. I want them to learn these things, but I don't want to tell them upfront, here's the vocabulary, we're gonna have a quiz on this. I would rather them learn it authentically by studying cool stuff about their home, by seeing how do the volatile organic compounds change in your kitchen if you're cooking. I think that's something students would find interesting, and they can learn about all these techniques on the side. I also really like DIY projects because students feel like they have a handful. You know, it's a very subtle difference, but they're not buying a pre-made, they're kind of connecting the sensor themselves. It's very easy, right? You just need a cable, but it gives them a little bit of a more engagement with how to take measurements. And I also think this project is very differentiable. For example, once students get comfortable, they can do something more with it. They can make, for example, a speaker that connect a speaker to the Arduino. And if the temperature exceeds 30 Celsius, sound goes off, for example. They can make it play different tones of sound depending on how hot the room is. There's a lot of differentiation you can do. Lastly, shout outs first off to my wife, Helen. Muito obrigado, amor, te amo muito. E também para Arthur, eu te amo muito. That's our family right there. Thank you also to my parents who are currently watching over Arthur right now. Thank you all so much. Love you all very much. Also to David, my lab mentor, he's been awesome. And Mara, she's taught me so much biology. Mara is so much smarter than I am, so thank you. Also, Megan Hill for running the program, Dr. Boyan Chin, excellent feedback. Dr. Clara Eichler, you were super helpful for inspiring this project, and Dr. Megan Lott for giving me this idea. Thank you so much. Any questions? Yes. Yeah, so after the question was, do they see each other's presentations? Yes, that's part of it takes a lot of time. So if you're a high school teacher doing this, those presentations might take a whole class period, or sometimes a class and a half. But I think it's absolutely worth it so they can see what they've done. And ideally, even if two groups do this project, their results will be different because they'll ask completely different questions. Yeah, great. Other questions?
Backwards, how do you navigate labs? But people where they really feel like these are real numbers, digits, there must be a challenge.
Yeah, it's a great question. So some of the numbers you can fact check fairly easily, like temperature and humidity. There's some local variations, but temperature can be measured. Most people have a thermostat in their house. But the other things like ozone layers, they can check with the ambient ozone of the day on Google, but there's still going to be some variations in that. I think it's really important to have students try to check these things as best they can. But yes, you just have to tell them these probably are not 100% accurate. This is a consumer brand chip. This is not the most accurate tool available. Thank you. Other questions? Thank you.
And last up, we have Carlos Rodriguez.
Hello, everyone. My name is Carlos Rodriguez. I'm a biology instructor at Wake Tech Community College. My lesson plan shows students how to use DNA techniques to identify fungi that live in indoor environments. The plan was developed from Gavin Duffy's doctoral research conducted under the guidance of Dr. Nicole Rocky at PreMiEr. So Gavin's project investigates viruses that live inside fungi that could potentially be used to control undesirable fungi that live in indoor environments. My lesson basically focused on the first stage of this workflow in which the fungi has to be identified with DNA-based methods in order for researchers to choose the best candidate that could be the host of these viruses. And the target audience are community college students that are currently enrolled in general biology, microbiology, or biotech courses. The lesson is divided in 3 and 4 segments. So lesson 1 basically introduces the students to background information on microbiomes and environmental parameters that enhance the growth of molds. And here you're having like a view of what is the information that is covered in that first lesson. And after the background information is covered, then lesson 2 introduces the students to the actual molecular tools or methods that are used to identify fungi. What you're seeing here is like an overview of the workflow that involves getting these fungi isolated, getting their DNA interrogated in order to check that DNA in a database and then identify the fungi. So this could be kind of complicated, but the idea here was to design lesson plans that could guide the instructor to be more effective in teaching this. So all the content of these lessons have features that support teaching and learning. What are these features? So every single slide in these lessons have guided step explanations and breakdowns of the lessons. Also, there are infograms included in the presentation that could be broken down systematically by the instructor using the magnifying tool in PowerPoint that summarizes very complex information in a single visual. And also in the lessons, there are hyperlinks that can allow the fast and easy access of resources without interrupting teaching. That also includes interactive simulations that further expand the explanation on the technical parts of the methods. Each of the lessons also supported with study guides, worksheets that enhance the learning and also facilitate the assessment of by the instructor. So in lesson 3, this is the part in which the students are going to be working actually with real data. They're going to use data of DNA sequences that are signature elements to identify different microbes. And they're going to use a program that is available online to analyze these sequences, compare them, and then construct tree diagrams that are going to assist in the identification of unknown organisms. In order to facilitate the teaching of this part, the instructor and also the student is going to receive a guided step-by-step explanation, click by click, so they can easily load the sequences, analyze them, and then construct the phylogenetic or genetic trees with this online tool. The idea is that the students are going to eventually identify an unknown bacterial isolate and compare it to a strain that is resistant to antibiotic that naturally makes a path for discussing the importance of antibiotic resistance in real life settings. There's a 4th lesson, which is entirely focused on the analysis of indoor fungi. Here, the students are going to work with DNA sequences that are DNA barcodes for indoor fungi and do the same workflow. They analyze online the sequence, they compare them. That comparison leads to the construction of another tree diagram that is going to reveal the identity of these unknown sequences based on their closest matches. So at this point, the discussion starts with an example of this fungi, Trichoderma, which is a natural fungal predator which can control the growth of other fungi. And that opens the door to explaining the role of these fungi as biocontrol. This fungi in particular has been in real life being used as biocontrols of pests in agriculture. And then from fungi attack, we move into intracellular viral parasites. So fungi can be affected not only by other fungi that parasite them, but they also have internal parasites, microviruses. So these microviruses, instead of killing instantly their host, they remain living long-term inside the viruses, and they weaken, debilitate the fungi, and prevents its growth and maybe the disease that they can provoke. And this is proof of principle that microviruses could be used as biocontrols. There's an example described in the literature on a microvirus called Cryphocteria hypovirus, which can be used to control the chestnut blight. And then using that as an example, there are other elements in this lesson in which other fungi can be discussed in different contexts with respect to how they impact human health, medicine, biotechnology, how they are used in space research and industrial applications. So the workflow here in this final lesson then leads to connect a career pathway with the research in microbiome to real life job opportunities. The method that they learn to do here or are exposed to are connected also to academic opportunities that are available in the local community college environment and the local work environment. So finally, I want to acknowledge and thank the Rocky Research Lab. Also, special thanks go to Gavin Duffy and Leslie Allison. I really enjoyed working with you. Thank you for everything you showed to me. And my special gratitude to Dr. Megan Hill for coordinating all this experience. Thank you. You have any questions? Well, that's kind of related to my background in microbiology. Oh, okay, so how did it get access to the?
Yeah, so you mentioned that like the lesson plans can be shared with communities, so other teachers, other professors can use it.
Yeah, yes, they can be they will be available for teachers. Yeah.
And then the teachers are coming to Cyrus here, which is their conference where they can then hand out their plans the teachers and start the data. And Cyrus it's the science.
Yes. So the question is, how much time is going to take to implement in the classroom this lesson plan? Well, this can be implemented in, I think, in 1 hour, 15 minutes lecture and a 3 hours lab in the context of community college classes. That's the target audience. But this lesson plan can be used in discrete units. The 4 lessons must not be used entirely. So since every single slide has its own explanation, so instructor can pick the slides that are useful and they don't have to figure out how to explain them. And that is going to make the task of putting this together into a new lesson more easy. So also the data sets have been already curated, proven, and they reliably function in the analysis. So the instructor doesn't have to spend time in doing data sets. That job has been already done in advance. They just upload the sequence along with the students, do the analysis, and they just get into interpretation. This is basically like computer lab work. It's not wet lab work. So the good part of this is that it doesn't rely, the implementation doesn't rely on the acquisition of reagents and supplies that are then spent. So you have the free online resource to do the analysis, you have the data set, and this can be run like indefinitely. Sure.
All right, we are going to move into a break. We'll be back here at 10:45, but before you leave, I just want to say thank you again to the teachers. Working with you has been such a pleasure, and I feel genuinely just so inspired by all of the work that you guys have done this summer. And thank you again to the mentors who invested so much time and energy into helping them succeed. And with that, come back at 10:45 for the REU presentations. Thank you. Yeah.
Hi, all. Welcome back. We're excited now to kick off this part of the symposium where we introduce our REU students, and they let us know what they've been up to this summer. So for those of you who might be new to the REU program, this is an overview of our structure. The students joined us here at UNC for 3 days for what we call our boot camp or orientation period. We heard from many of the faculty within PreMiEr and got a crash course on all of the things that the center is focusing on and advancing. Students then went off to conduct mentored research at one of the PreMiEr institutions with 8 of our different PreMiEr faculty. They were mentored by graduate students, postdocs, and supported by you all in the center. The students just returned here to UNC this week on Wednesday so that they could prepare their final presentations, get and give feedback. And in addition to this formal programming, I'm really excited to see that the students have made the best of their summer. I want to give a large thank you to all of the folks within PreMiEr and beyond who have provided mentorship, training, and support to this cohort. We appreciate all of you. You build a welcoming and supportive scientific community that lifts all of our scholars at all stages. So thank you. With that, I'd like to introduce our first REU speaker, Edward Zhuang from the University of Texas at Austin, who is starting his senior year and studying biomedical engineering this summer. Edward was working in the Wisner lab, and I'm excited for him to tell you more about hydrogels.
Hi, everyone. I'm Edward, and I'll be presenting on the biological compatibility of encapsulated probiotics and hydrogels. So the issue that I've been looking at is that hospital sinks act as a pathogen reservoir, and these pathogens form biofilms in sink drains and P-traps that provide protection from cleaning and disinfection, and also act as a supply of bacteria after most are washed out. And because of this, they can end up migrating and spreading to surrounding surfaces through aerosolization and droplets dispersion, and end up infecting patients. And some of the specific pathogens that are used is Pseudomonas aeruginosa. So some of these current disinfection methods, they do work, but they do have some limits. For this case with these biofilms, these disinfection methods aren't able to penetrate the deep biofilms that they form. There are some physical barriers involved, safety risks, costs and operational disruptions. And one of the major issues as well is antimicrobial resistance, where all these disinfection methods over time could lead to resistant strains evolving and surviving. So what am I exactly looking at? I'm looking at a synergistic approach between both probiotics and hydrogels. So the first thing we're looking at is the probiotic community itself. This is composed of beneficial bacteria that can form their own biofilm and outcompetes these pathogenic bacteria, and also break down the pathogenic biofilm as well through competitive exclusion and provide their own continuous presence while also being non-toxic to patients and staff. And the other section of this approach is the hydrogel delivery method itself. So hydrogels themselves are a hydrated polymer network, and they can encapsulate these bacteria, provide storage and protection, while also being very biocompatible and tunable due to many different methods of creating them and what sort of materials you use to make them. And this leads to my overarching research goal for this summer, which is to evaluate the biological compatibility of these 2 different methods. So first off, with the probiotic community, this was provided to us by the Venturelli Research Group, and how they evaluate it is to look at both the reduction of P. aeruginosa in both experimental and low nutrient conditions. Low nutrient conditions, in this case, is a 1/5 concentration of R2A, which is where these bacteria are able to thrive and grow in. And we ended up getting this 5-member community that had a 2.5 log reduction in normal R2A and a 0.5 reduction in 1/5 concentration R2A. So the first thing I'm looking at is our experimental setup with the hydrogels themselves. The 2 gels that we used are a sodium alginate or SA hydrogel or a sodium alginate with crystal nanocellulose or SAC&C hydrogel. And the reason behind this crystal nanocellulose is that it exhibits a 10-fold mechanical strength, which is why I wanted to see if this would have any effect on the biological compatibility between this and the probiotics. And we're comparing these to a positive control of just bacteria in solution and a negative control of just plain media. And the process of making them, as you can see in the diagram, is pretty simple. you encapsulate these probiotics in the stock solution of hydrogels, and then you spray with calcium chloride so that they can ionically cross-link together. And then they form these polymer networks that you can see on the bottom here. So one of these biological compatibility tests is bacterial release. We want to see if these hydrogels can release release the probiotics healthy and able to sustain a good population over time. In this process, we submerge these hydrogel samples in this low nutrient media. And every 24 hours, we remove the media, rinse out the wells, and submerge in fresh media to then evaluate the next day. And then this removed media, we then look at the optical density in order to get a gauge of like what the bacteria concentration looks like. And from our results, we see that both gels indicate that they have a very, very good sustained release of bacteria. Within the first 2 days, we see that major increase in bacterial release. And throughout the 7-day process that we tested this, we were able to see that the released bacteria are able to sustain a population. And we hypothesized that this released bacteria, because we're replacing this media every single day, they're able to repopulate the well plates and from that fresh media as well. And the second part of this biological compatibility test is looking at the biofilm formation, because we want to make sure that these released probiotics, that they can form a healthy biofilm and be able to combat pathogenic bacteria in the future. So this is a relatively simple process where we submerge the hydrogel samples in the low nutrient media. And at each respective time point that we checked, we would dump out all the gels in the media, rinse out the wells with PBS in order to get rid of loose bacteria, and then stain the leftover attached bacteria with crystal violet, and then measure the absorbance of that crystal violet. And from our results, we can see that there's a significant improvement in biofilm formation compared to the positive control. And each day from 1, 2, 3, 5, and 7 days all show a much higher formation of biofilm. And what we're seeing is that the gels are constantly providing that consistent source of bacteria from what we saw in the release test so that they're able to constantly replenish that biofilm. And to provide more context, when we look at the raw crystal violet absorbance values, we can see that we have the greatest biofilm formation in the first day. And over time, this does end up decreasing over time, but we hypothesize that this could be due to the nutrient being depleted over time, because with each of these well plates, they're sat for their respective time points and then analyzed. And over time, as the nutrients are getting used up, attached bacteria from the biofilm end up detaching and getting into the planktonic solution itself. And as you can see with day 3, we start to see some of those differences in that drop where the sodium alginate gel actually decreases a little less than the sodium alginate CNC, but the positive control does end up dropping a fair bit. And so overall, these hydrogels show a very promising delivery method of these probiotics. Both show very similar levels of biocompatibility, so we can evaluate the trade-offs there for the sustained release and the increased biofilm formation, all of which are very positive things to see. And from this, we can also evaluate like when these probiotic communities could be established in a real world scenario, because as we saw that peak biofilm formation in day 1. So that means like if we were to apply this to say a P-trap, and we'd be able to establish our probiotic community pretty quickly without interruption of services. And so future work for this, we would want to evaluate how well these gels and their encapsulated probiotics are able to reduce P. aeruginosa, and then also evaluate if these are able to protect against disinfectants to make sure that these are able to survive and continue creating their own biofilm, and then test it in real world scenarios such as the beach. I'd like to thank everybody that I've worked with in the Wisner Lab, PreMiEr, and everybody in the REU cohort. A huge shout out to my mentor, Kobe. It's made this summer very great in the lab, and I'm very grateful for this whole entire summer experience. Thank you all for listening.
So you talked about all of this before, using the hydrogel to help fight Pseudomonas aeruginosa. So like in that case, would you like kind of applying like a gel like thing to like a biofilm Pseudomonas, and then having all the probiotics kind of like fight it out with the Pseudomonas?
So the question was, how would we apply the gels with probiotics to actually reduce P. aeruginosa in the future? So some thoughts I've had is that we would establish a biofilm first and then then introduce the P. aeruginosa, or we could also just establish it, like you were saying, by putting the probiotic gel with an already established P. aeruginosa biofilm. But that's also still up to discussion, like how we would go about checking how it would reduce. We also want to look at levels of P. aeruginosa biofilm on its own in these low nutrient media, just to have a comparison with, like, does the probiotic form a better biofilm than the P. aeruginosa on its own? And those are all things that we want to evaluate in the future.
Liesl Allison, who is joining us from Chatham University. Liesl is studying cell and molecular biology. She'll be starting her senior year. But this summer, we've had the pleasure of learning from Liesl as she wrangles fungi, mycoviruses, and all of the above in the Rocky Lab. Liesl?
Hi, I'm Liesl, and I will be presenting my work establishing transformation protocols for study of mycovirus impact on built environment fungi. So mycoviruses are fungal viruses that infect and use fungi as their hosts. And whenever mycoviruses infect fungi, they can alter the host expression, specifically increasing or decreasing pathogenicity or virulence, or increasing and decreasing growth rate. But there is minimal mycovirus identification and characterization, meaning that there are lots of mycoviruses we have not yet isolated and lots of things we do not know. But one of the things that we're interested in is using mycoviruses as biocontrol agents against fungi, specifically mycoviruses that induce hypovirulence in the fungi. So one of the overall goals of PreMiEr is to utilize mycovirus hypovirulence as a targeted biocontrol agent against fungi in the built environment. So in really simple terms, to do this, we would isolate a sample or samples from built environment, identify any mycoviruses that are infecting the sample, identify the virulence pathway that the mycovirus is altering, and then deploy the mycovirus as a fungal biocontrol. So at the beginning of my time here, I started working on some samples that had been collected from the PreMiEr House and the Wilkinson building. And when they had been collected, they were plated, and that created complex plates with a whole bunch of different samples on them. And so then I worked to culture these samples with an end goal of getting a bunch of individual isolate plates that only had one sample on them. And these environmental isolate plates they're often not model organisms, but we hope to eventually use them as biocontrol agents in the built environment. So here we have 3 yeast samples that were isolated from these areas. And in the middle, we have the ITS PCR bands that indicate that they are fungal samples. And since we isolated these, this was a motivation for us to develop a protocol for yeast transformation. But to be able to study how the mycoviruses impact the host, we have to be able to study the effects that they have on the host. But because of the lack of knowledge, the extracellular entry method that microviruses use to enter their host cell is unknown. And this is where transformation comes in. So transformation will allow us to insert a known mycovirus sequence into a fungal host and then hopefully it will be expressed, and then we can observe the impacts on the host. So a quick overview of transformation. The cells are prepared to make them competent, which basically means that they are kind of go into survival mode. They're like on the brink of death, basically. And so then whenever they're exposed to the plasmid, they are more likely to uptake the plasmid because they want anything that could possibly give them any better chance of survival. And so then we introduce an environmental stressor that will kind of scare them, and then they take the plasmid. And then they're plated on selection media, and if the transformation is successful, theoretically, the colonies that grow should be transformants. So to go in a little more, there's many methods for transformation. Generally, it's been done with filamentous fungi, which one of those methods is Agrobacterium tumefaciens mediated transformation, or AMT. And this has been being done by Brianna Jameson at NC State, and I was actually able to go and observe her doing that transformation. And so then I aimed to do the transformation with yeast. So I used the lithium acetate transformation. So I adapted the lithium acetate transformation from a couple different protocols. So I used Saccharomyces cerevisiae and grew it up in a culture, and then diluted it down to a set concentration so that we could then incubate it and stop the cells when they are in their exponential growth phase for the transformation. And then the cell pellet was washed and the transformation mix was added, including an empty plasmid, just as our first, to start out. And then we heat shocked the cells, which is our environmental stressor, to get them to uptake the plasmid, and plated them on selection media. I plated full concentration and then at a 1:10 dilution. Incubated them, and then hopefully would identify transformants. Unfortunately, we had no identifiable transformants recovered. So this was our control plate, and you can see the S. cerevisiae has grown all over the control plate, which shouldn't have happened because the selection media should select for a specific gene within that's added to the plasmid specifically to make it grow or not. And then this was our transformer plate, which looks the exact same as the control. Still not very good. And the expectation would have been that there were a whole bunch of individual isolates, isolate colonies that would have shown. And this happened because we used the wrong selection media on accident. So, oh well. But so in conclusion, the lithium acetate transformation, I still think that it remains a viable option for mycovirus insertion because the cells survived and they could have been transformed. We just don't know that. So if we had used the right selection media, it could have worked. So there's no reason to count it out yet because we just don't know. And then because we were able to isolate the environmental yeast isolates from the built environment, which are not really that common, we could use this lithium acetate transformation with those isolates in the future, and then hopefully go from there. So if I had more time, I would obviously redo the S. cerevisiae transformation on the appropriate selection media. And then if that were to be successful, then I would insert a known mycovirus sequence into the plasmid and transform it. And I would like to thank everyone in the Rocky Lab, specifically Gavin Duffy for being a great mentor, and our collaborators at NC State, everyone in PreMiEr, and then of course the REU cohort. This has made the best summer ever. Thank you. Oh, okay. I honestly hadn't thought about it that much. I know that they do a lot of I don't know how to word this, but they connect a lot through their hyphal tips. So I feel like that would be a really good way for them to share viruses, or not intentionally share them, but for the virus to go, I'm gonna hop over there. Yeah. Yeah, that would be mine. Because they do a lot with their hyphal tips. They can. So in at the beginning where I showed the three isolates, the 3 yeast isolates, the like pinkish orange one, if we set that aside, the other 2 we actually think were dimorphic yeasts, which you could kind of see in the picture. So they were growing kind of like the pinkish orange one kind of looks a little bit bacteria-ish, but then they also were like kind of looked filamentous and were like growing out. So they can do both. They can go back and forth and do both.
Next, we're excited to welcome Jessica Chin to give her presentation. She's joining us from the University of Massachusetts at Amherst, where she's starting her senior year studying microbiology and environmental science. She's been working this summer in the Rawls lab, and we're excited to hear what she's learned about growing and surviving in the aerobic environment.
Hi everyone, my name is Jessica, and today I'll be presenting on my past research in the summer in the Rawls lab on identifying genetic fitness determinants of Fascicula vulgaris in aerobic built environments. So firstly, there is a crucial intersection between human gut microbes and the built environment as a whole. All human gut microbes are acquired from the environment, and more specifically, surfaces of the built environment are likely to be a major reservoir that plays a role in that transmission. And we want to learn about this because we see that a lot of humans will live in a lot of built environments. Humans spend up to 90% of their lives indoors, so it's crucial for us to understand our built environments and our indoor microbiomes as a whole. So this poses the question of, how do human gut microbes survive in the built environment? So in order to answer this question, we wanted to focus on one specific bacterium, which is Fascicula vulgaris, otherwise known as PVU. It is a prevalent human gut bacterium commonly found on built environment surfaces. It is normally residing within your gut microbiome and is normally digesting fiber. And it is an aerotolerant anaerobe, meaning that it is an anaerobe, but is also capable of surviving brief oxygen exposure. This trait allows it to be one of the most abundant species within your gut microbiome, but it also allows it to survive on surfaces of the built environment, such as refrigerators and toilets. So this study done in 2013 shows that when a refrigerator and toilet surfaces were swabbed, a good amount of the gut microbiome that were found on those surfaces ended up being PVU. Additionally, PVU is also a species that have a lot of extensive gene transfer, extensive gene transfer capability, which allows us to make clean deletions pretty easily and then also genetically manipulate it as well. So this is the brief project overview. So our first step is to screen the PVU transposone library for mutants that show differential survival on PVC and HAP plastics in aerobic and anaerobic environments. So to break it down a little bit, we're going to screen the PreMiEr transposon library, and a transposon library is a library of mutants that have random insertions called transposons that essentially will jump randomly into different genes and then disable those genes, preventing them from, for example, transcribing a protein. And then we want to screen those on our selected conditions. So in this case, we're selecting them on different types of plastics and also in aerobic and anaerobic environments, and then seeing whether or not deleting that gene will, or disabling that gene will either enhance or enrich or deplete their survival within those different conditions. And we picked the PVC and HDPE plastics because those are plastics that are commonly prevalent in plumbing, which is one of the PreMiEr's built environment interests. And we chose them based on our collaboration with the Wisner Lab. And after this, we know that making a transposon library is only a quick overview of our data. So we want to make clean deletions for those genes of interest in order to solidify and confirm our results in vitro. And then lastly, we want to screen those clean deletions, and then we want to screen those by making direct competition assays versus basically competing wild type and mutant survival on those different types of plastics and in anaerobic and aerobic environments. So this is the detailed transposon library plastic assay that a previous Rawls Lab undergrad, Ethan, conducted. And he basically just screened the transposon library that was made on the different types of plastics and also on different anaerobic and aerobic environments. And this was the data that they yielded. So to quickly break down the volcano plot, we were specifically interested in the 24-hour aerobic PVC versus 0-hour PVC. On the left side, you can see those quadrants. Those quadrants are genes that were depleted in 24 hours aerobic PVC. And then the quadrants on the right are those that were enriched in 24 hours aerobic PVC. So we were interested in the top right, which basically shows genes that are both statistically significant and then also surviving better on aerobic PVC when that gene is knocked out. And we saw that there were a few different correlations. So for example, there are a few mutants that were within the same pathway. It was 3980, which is up there, 3981, which is down there, and then 3986, which is also down there. They're all involved within the same acid path, in the same pathway. However, we saw that only one of those genes was in the quadrant we're interested in. That's both statistically significant and survives better on aerobic PVC. So we decided to make a clean deletion of 3980. And you might be wondering, what does 3980 do? And what is that pathway? So 3980 encodes for glutamate decarboxylase, which is an enzyme that plays a key role in the glutamate acid resistance pathway, which allows for gut microbes to survive within your gut microbiome. So we were interested in making a clean deletion of that. And then we ended up making the clean deletion. Unfortunately, the process was very long. That was the majority of my summer. So if you guys have any questions on that, let me know. But unfortunately, for time's sake, we weren't allowed to, or I didn't have enough time to present it, but definitely ask me either at the reception or at the end of this presentation, because it was a very cool and fun experience to make. But yeah, so we made the clean deletion, and we wanted to see whether or not deleting that gene influenced its growth compared to wild type, because we would need that information for later assays. So we conducted a growth curve, and we saw that they grow pretty similarly to one another. But we see that there is also a potential earlier log phase onset. You can see that around 10 hours, the orange line is the mutant of the 3980 deletion. And you can see that it might have a slightly earlier log phase, meaning that it might be growing a little bit faster. But we definitely do have to repeat this just to confirm that it is a trend that we can see in different biological and technical replicates as well. So after we made that deletion, we now wanted to go on to our last step of the assay, which is to test it on the culture-based direct competition assays and see whether or not deleting 3980 allows for that bacteria to outcompete wild type on survival on aerobic PVC over 24 hours, which is, if you remember, it's the transposon library data that we verified in the past slides as well. So we just want to verify that transposon library data. And so for our first run, we saw that the mutant of 3980 may survive better on the aerobic PVC plastic. So basically, you can see that the bars and the y-axis are the percent mutant, and then everything that makes up wild type is reaching up to 100. So comparing both the PVC aerobic and anaerobic data, aerobic being around 79%, and then anaerobic being around 77%, you can see that the mutant widely outcompetes wild type However, we also can't say this for sure because we had a few issues with the input data, so we couldn't solidify our starting concentrations of the wild type and of the 3980 deletion. So we'll definitely be repeating this again in the future to just verify that these results can be confirmed once we know the input data of how much bacteria we had from each of the strains. And then we also wanted to compare them based on the aerobic and anaerobic side for the PVC plastics. And you can see that there is a very small difference. There's around a little bit more than 1% difference between the PVC aerobic versus the PVC anaerobic. So we can't confidently say again that the PVC aerobic plastics are enriching the survival of the 3980 deletion. However, it is good preliminary data to go off of for future assays. So as a whole, these are conclusions of our past research. So we saw that the mutant of 3980 and wild type grow similarly, though there may be a potential earlier log phase. The mutant 3980 may also be slightly enriched in the 24 hours aerobic PVC, but again, we need to solidify our input data and then do another run of the experiment. And in general, additional testing is needed for us to continue in the future to finalize our plastic protocols for assessing the wild type and mutant competition growth. This includes finding an ideal concentration of the bacterial culture or adjusting the pipeline of the 3980 mutant grow out. We did repeat this experiment again within the last days of the last week. And unfortunately, we did change the way that we inoculate our bacteria. So we didn't have a high enough CFU to screen the bacteria in order to get a good ratio of wild type versus mutant. So we'll definitely have to be increasing the amount of bacterial culture that we are adding before mixing in with our pellets. And then we also need to repeat the growth curve to see if we need to adjust the pipeline of how we are inoculating our bacteria and what time we inoculate our bacteria and how we subculture it for potentially making 3980 mutant and the wild type to grow so that both the input data are within the same bacterial CFU range. If you remember before, there were also a few mutants that stood out to us in the transposon data. Specifically, PEG 3979 was both in the enriched and also statistically significant quadrant that we were interested in. It wasn't within the same pathway as 3980, so this is definitely going to be something that we'll want to look more into in the future. And then also 3981 and 3986 were within the same pathway. They just weren't statistically significant genes. So we'll be interested in seeing whether or not knocking those genes out will have the same impacts as 3980. And then doing further comparisons with HDPE and PVC plastics. We want to repeat this assay with HDPE plastics and seeing how changing the different types of plastics will change the wild type versus mutant ratio. And then further analyzing how this can reflect on the bacterial growth and household appliances and daily use plastic products. So I just quickly want to say thank you to the Rawls Lab for having me this summer. Special shout out to my two mentors, Jess and Lisa, for being amazing mentors and being a great support system for me. Thank you to PreMiEr, thank you to the Wisner Lab, and thank you to the 2026 REU cohort who have kept me mentally sane throughout the summer. But yeah, thank you for listening.
Covered in brine, organic films and oils and all this like that. How do you think that might affect your experiments if you incorporated the complex organic films onto the plastics?
I'm sorry, can you repeat that one more time?
Sure. So all surface forms, but covered in brine, like oil, detergents, and whatever. If you were to incorporate that into your experiment, actually putting your plastics some layer of these organics, how do you think that might affect your experiment?
Yeah, so the question was asking whether or not the different types of grime or different types of already existing contaminants on the built environment surfaces will impact the results of our plastic bead assays. And I will have to think about that. When we did the plastic bead assays, we did have to rinse and clean the pellets. So I'm thinking maybe with the grime, or like with the stuff that's already existing on the built environment surfaces, it'll probably impact our data in terms of like what other bacteria is already on the surface, which will impact the way that we screen. Because when we were screening, we basically picked 24 different clones of each different surface, and then we see whether or not it's wild type or mutant via PCR and then via gel electrophoresis. So I'm assuming that if there is any impact of those different types of surfaces of like already pre-existing bacteria or pre-existing grime, that will reflect on the screening because the primers that we're using only select for PVU. So it'll only tell us if it's PVU and if it's wild type or mutant. So in terms of screening the assay, I don't believe it'll have as big as an impact on the assay. It'll just mean that we need to take more clones for screening so we can account for, other different things that are impacting the bacterial growth on those surfaces. I don't know if that answers your question, but okay. Yeah. That was under anaerobic conditions. Mhm. Normally PVU is found in fecal matter. So when I was in the Rawls lab, I did a lot. Dr. Rawls gave really interesting input on how he believes, and a lot of other scientists in the gut microbiome industry believe that when humans are born, they're born gut-free, not gut-free, germ-free within their gut microbiome. So I'm assuming there's one specific route, which is a fecal oral route, which is normally probably where a lot of bacteria from like different environments will go into your gut microbiome. And then additionally, you'll probably have PVU transmitted from like your mother, because PVU is also another species that is highly prevalent in the within birth. PVU is normally also transmitted from the mother down to the child as well. So, and then, yeah, so normally PVU does live in the gut, and it's also found a lot on surfaces. So I'm assuming that there is a lot of different ways for us to pick up PVU, but it is normally found in fecal matter as well. So I'm assuming the main route is the fecal oral route. I didn't do too much research on different host species, but because it is, so PVU normally allows for like fiber breakdown within the gut. So I'm assuming other different species that require that type of mechanism will have PVU, but I'm also assuming there are other microbes within the gut microbiome that perform that same mechanism. So I can't confidently say if other species kind of have that same PVU prevalence within the gut microbiome, but I think that's something to look into in the future as well.
Introduce. Oh, is this a bonus slide? Oh, the supplemental info. Find Jess at lunch if you want to talk about these. Okay, Kathy Go, who is joining us from the University of California at Berkeley, where she's beginning her second year studying microbial biology. And this summer has been working in the Newman lab to generate a new bacteriophage. Kathy?
Okay, so hello everybody, my name is Kathy, and today I'll be going over my summer project, which was the whole genome synthesis of the Pseudomonas aeruginosa phage, PE3. So introduction. As many of you guys know, there are a lot of infections that are commonly acquired in hospitals, and at the root of these infections are usually a group of pathogens known as escapee pathogens, with one of them being Pseudomonas aeruginosa. And this causes things like lung infections, UTIs, bloodstream infections, and more. PreMiEr sought to answer the question, how can we better combat Pseudomonas aeruginosa in hospitals? And one idea was by engineering the natural-born bacteria killer, which are bacteriophages. So my summer research question was, how can we design and create a synthetic bacteriophage to better combat Pseudomonas aeruginosa? And it can be broken down into 3 main steps. Number 1, making the phages. Number 2, testing different variants of phages. And number 3, quantifying the results to see which phage can eradicate Pseudomonas aeruginosa for the longest time and the best possible way. And the step in which I joined in was step number 1, which is actually making the phage. So how do we make a phage from scratch? First, let's talk about why we chose PE3. PE3 is a recently isolated phage that has a genome length of 43.58 kilobase pairs, and it is lytic, meaning that upon reproduction, the bacteria will be killed and lysed. And the main reason why this phage is great is because it has a lot of room for improvement and a lot of room for being engineered. There has been studies that show that upon the reduction of 12 different genes, this bacteriophage can actually function pretty normally. So we can reduce the genome to allow for areas of adding our own genes of interest and other payloads. And directions that we can take this is by expanding the host range. It can only affect 7 out of 28 clinically relevant strains. So what if we could engineer it to infect all 28? Like, that would be really cool. And also another thing is that Pseudomonas aeruginosa evolves resistance really fast within 24 hours. So what are the kind of genes that we can possibly add to disrupt their quorum sensing or their ability to persist and develop resistance? And now let's look at how we actually broke down the genome. So given the full phage genome, we first cut it up into 7 mini chunks, with each mini chunk having 4 fragments, for a total of 28 fragments. And all of these individual fragments were stitched together with a technique known as Gibson assembly. So what is Gibson assembly? Gibson assembly is a way in which we can join different DNA fragments in a one-pot reaction within like an hour or so. And how this works is that, as you can see in the diagram, given fragment A and B, we can, in our computers, add a section in which they're complementary to each other, so kind of like sticky ends. And then once we put these two DNA fragments into the reaction pot, there will be a 5' end exonuclease, which will chew back the 5' end, revealing the two sticky fragments. And those two will anneal together. And to fill in the gaps, there will be a DNA polymerase as well as a DNA ligase to conjoin the empty spaces, essentially. And so this is kind of the essence of all of our experiments, pretty much. And so once we put together each of the fragments together using Gibson assembly into our mini chunks, we can then do more Gibson assembly and put all the mini chunks into an even bigger yeast backbone, which is the PRS415. And this is how you make a phage, essentially. One way to do it, essentially. And once we have the entire phage genome created, we can use an in vitro transcription translation system to transcribe all the genes and make all the proteins. And hopefully, if all goes well, we will be able to see the phages self-assemble in our labs. And one really important thing that we refined and developed through this summer is how to screen. What does screening mean? It basically means like we want to verify that when we put together the genome, it's actually correct. And we do this through colony PCR as well as site-specific PCR. Colony PCR will amplify the entire insert region, so all of the assembled gene fragments, whereas site-specific PCR will only check the most newly attached fragment. So these two ways are kind of our ways of checking. Now let's move on to results. I was tasked with making mini chunk 5 through 8. So as you guys can see, it's pretty much, we start off with the backbone, a linear backbone plus fragment 5. We add on fragment 6. We use PCR to cut that circular plasmid into a straight linear fragment, add on 7, and then same process, and then add on 8. And so first, it's really important that we verify our starting materials, which is the already verified PUC19 backbone plus fragment 5, as well as the purified fragment 6. And as you guys can see in the gel, the length is correct and that they're all consistent at 2.3 kilobase pairs. And now we can start by adding fragment 6 to fragment 5. So after doing this using Gibson assembly, we did our 2 standard screening procedures, which was the colony PCR and SSPCR. And the takeaway from these 2 gels is that something may be wrong, because it seems like it's around 500 base pairs short for both of the gels, which is why at this point we sent it out for sequencing. And the results came back and it was fully correct, which is what really confused all of us, because what was that discrepancy? And we don't really have a very well developed hypothesis, but one issue could have been that I used the wrong extension time for the PCRs. I set it as 30 base pair per second where it was supposed to be 1 kb. However, this doesn't seem too likely, because if this was the case, there shouldn't be bands at all. So this is still kind of a work in progress, an area in which we're trying to figure out why there was that discrepancy. But it must have been something in the PCR. So after getting these confirmed results, we continued onwards by adding fragment 7, Gibson assembly, same drill, and then got the colony PCR, site-specific PCR. And the conclusion from these two gels is that something, like, it seems like there's some issue with the PCR that's still occurring, and there's still some issues with the length of the bands. But despite that, we continued onwards, and we linearized using PCR. We turned the circular plasmid into a straight DNA fragment, and we amplified it. And then we saw 3 lines with 3 different bands, which is really weird. So one way we troubleshot this is by increasing the annealing temperature. And what this will do in your PCR is that this will essentially allow your primers to bind to where you intend it to bind, and it'll increase the chance of correct amplification of your targeted sequence. And indeed, there was a huge difference in just those 2 degrees Celsius. The band that we were looking for at 6.3 kb is much more strong, and the other 2 off-target amplifications were much weaker. So this is actually a pretty neat trick to do with your PCR if that is an issue. And last but not least, we're adding fragment 8, and we did everything. I used the right extension time, and thankfully the bands were right this time, so it must have been something wrong with the previous ones, not too sure. But we can all conclude that it is really important to have the correct thermocycler settings when doing PCRs and when doing gels. And in conclusion, we built mini chunk 5 through 8. And hopefully in the future, we will be able to continue onwards to assembly of the full phage genome and also make more variants. And actually, like 5 minutes before my talk, I just got the sequencing results back, and it seems like everything is correct. So yeah, thank you guys for listening. Big picture and looking at genome, which species are for manipulation for your end goal? I think one of the most exciting ways in which we can manipulate the genome once we build it is by adding genes that change the tail fiber of the phage. The tail fiber is basically like an appendage that helps recognize different strains of the virus. So if there's a way in which we can kind of tweak it so that it recognizes more different strains of bacteria, that means that it can target and kill more bacteria, more types of bacteria, basically. And that's one way. And also, another thing is that we actually are already trying to build a variant with this gene called AIIA. And what this gene does is that it actually messes with Pseudomonas' signaling, and it makes it hard for the Pseudomonas to tell each other that there's a threat around. So basically, it disrupts the quorum sensing, and that will disrupt its ability to form biofilms and infect people. And so, yeah, and there's definitely many other ways, but those are the kind of 2 things that jump out at me. Questions? Yes. Yes. Yeah, so how we did it is that we did it in the computer. So when we made the genome, we added on a 30 base pair sticky N to each fragment so that when we put it in the reaction tube, it will automatically anneal together because they're complementary. So usually it's done via computer. Yep.
Mhm. Thank you. Any other questions? Oh. Okay. All right. Thank you, guys. Oh. Oh no. Nope. Hi. Hi, folks online. Welcome back. We're excited for our next set of presentations. Before we begin, I want to give a quick shout out to Dr. Kristen Landraville, who is going to move from the PreMiEr ERC to take a chair position at the University of Wyoming. We'll miss her greatly. Our REU students were able to work with her to think about the SEI implications of their work and then work with the RETs as well for their lesson plan development. We thank her greatly for this participation in these summer programs and also in our research broadly. If you'd like to sign an e-card for Dr. Kristen Landraville, you can use the QR code here. Just flip to the next E page and there should be a space to sign. All right. Up next, we'd like to welcome Evan Hankinson, who is a student from far, far away down the road at Duke University. He's studying biology, entering his senior year, and has spent the past 2 or 3 years working in just about every lab within PreMiEr. This summer has been working with Drs. Thomas and Bengal, and we'll tell you more about his bio-inspired nanoparticle research.
Morning, everyone here and online. So I'm Evan. I went into this summer planning to do some evolution research against nanoparticle surfaces, and as science tends to go, I ended up doing something completely different. And so this summer, I worked on designing bio-inspired nanostructures to enhance antimicrobial surface chemistries. And so to zoom way out from this, sort of why are we doing this? And as some other people have mentioned, hospital-acquired infections are a really big problem. As many as 1 in 10 patients in a hospital end up with a secondary infection from that hospital. And those infections are often transmitted from surfaces of medical equipment and hospital facilities. So our thought is that by modifying those surfaces in some way, we may be able to prevent the transmission of bacteria in hospital environments. And so one of the ways that we can modify a surface or a type of antimicrobial surface that's been used for a long time is silver. So as far back as around 750 BC, people were using silver as an antimicrobial on things like silverware, which is where we get the word from. And throughout history, it's developed even after the advent of antibiotics as a continued method of combating bacteria. And recently in the 2000s, silver nanoparticles have become something of interest for their increased activity against bacteria, especially within sort of like liquid environments, which inspired us to look at the potential of using these silver nanoparticles against bacteria on surfaces. So some of the previous research in the Thomas lab, and as well as the Graves lab over at NCAT, has studied how these silver nanoparticles interact with bacteria in solution. And one of the mechanisms that was proposed by them was that there might be a physical interaction, a physical threat that some certain shaped nanoparticles might pose to bacteria. So if you look at these triangular nanoplatelets, you can see that the edges of those might be able to sort of poke a hole into bacteria. And on a surface, those physical traits might interact with the chemical traits to enhance them or possibly provide increased barriers to bacteria evolving resistance. And so in order to test some of these silver nanoparticles on surfaces, I synthesized these silver nanoplatelets in the Bengal lab and inoculated a weld plate with these nanoparticles and allowed it to dry and settle into these nanoparticle surfaces. And then using a GFP transformed bacteria, I cultured those in what's called a semi-dry environment. So I placed drops of those bacteria onto these silver surfaces and allowed them to settle and dry overnight, and then resuspended them in media the next day and took fluorescence readings of the GFP to estimate the survival of the bacteria after that time period. And unfortunately, those silver nanoparticle surfaces didn't work. So you can see here that after the 24 hours had passed, there's not a clear effect of these silver nanoparticles. And so there's a few reasons that we think that might be happening. One of those is that silver nanoplatelets are notoriously flat on their sides, which means that there's not a particular physical threat when you place them down into a surface. And there might be other relationships with their persistence on the surface and with concentration that are limiting us in terms of what we could do with the nanoparticles. And so that encouraged me to look back at how we design a solid physical threat against bacteria. And one of the best examples of that is a cicada wing. And so cicada wings have microcolumn or nanocolumn structures on their surfaces, which are very good at poking holes in bacteria, and in fact work as excellent antimicrobial surfaces without any clear chemical interaction occurring. So in order to replicate these nanocolumns, I used a method called colloidal lithography, where I created a monodispersed layer of nanospheres on a silicon surface. And then using what's called reactive ion etching, I ate away at that surface using those monodispersed nanospheres as like a shield to form these nano spikes that can hopefully poke holes in bacteria. And so this is an image of my assembled nanosphere layers. And so by adjusting variables like drying time and temperature, the angle at which these were deposited onto the surface, and the concentration of them in our solvents, we were able to consistently form relatively even monolayers on our surfaces. And then after reactive ion etching, you can see that we created these like spike patterns across the whole surface to act as hopefully act as our antimicrobial. And this is a atom force microscopy image that shows the topography of the surface after etching. So there's a clear formation of these spikes. I want to note that these spikes, most of them are between 75 and 100 nanometers tall. And cicada spikes are usually around 200 nanometers tall. So spikes that we produced in this experiment were not quite so tall or consistent as a cicada wing. And so we then put coatings over these spikes. So some of our coatings were silver to sort of to show the relationship of like these antimicrobial chemistries that we had previously looked at with this surface. And then we also used an aluminum oxide coating. So we thought that there might be some interactions with hydrophilicity in these surfaces. And so aluminum oxide is strongly hydrophilic and lets us sort of understand how wettability properties might be influencing these surfaces. And so after a 48-hour semi-dry incubation on the surfaces, you can see that the rough or the spiked surfaces did not perform as we expected. So in the silver environment, silver is just such a strong antimicrobial surface that after the 48 hours, effectively all of the bacteria on the surfaces were destroyed. But in the silicon and aluminum oxide, where there's not that antimicrobial chemistry, it seems like the rough surfaces actually enhanced the survival of bacteria, which was not what we were expecting. But there's a strong sort of explanation for this, and that has to do with capillary pressure. So between these spikes, there's these like sort of nanoscale spaces where there's a high capillary pressure because the water is so close to the walls, and especially on our hydrophilic surfaces, you can see that water is likely being trapped and persisting on the surface, preventing the desiccation. Whereas on the not spiked surfaces that are hydrophilic, because the water spreads out really evenly, it evaporates quickly and the bacteria desiccate. So the aluminum oxide helped reveal that to us, which made us think, what if we incubated just 12 hours against bacteria to see if these might enhance just the silver surfaces? And it seems like, these are results from Wednesday morning, and I didn't have a lot of time to replicate them, but it seems like the rough surfaces do actually enhance the surface chemistry of silver, or at least how fast it kills bacteria, likely through this like wettability effect and persistent water on the surface helping to release ions, as well as this increased surface area effect where there's more silver available to attack the bacteria at a given time. So in summary, silver nanoparticle surfaces were limited for the applications that we were looking at, but the colloidal lithography surfaces, while they don't necessarily yet provide a strong physical threat yet, the surface area effects and capillary pressure might be enhancing the surfaces, especially with pre-existing antimicrobial chemistry. So thank you, guys. Open for questions. Yes, I will.
Looks like you added coding for all of the situations, right? What if we didn't need to, like on the cicada wing, I guess, have kind of antimicrobial coding, or is it quite outside?
Yeah, so the question is that, or sort of what would happen if we didn't add coatings to the nanocone surfaces? So I do want to just clarify really quickly. We didn't coat the silicon surfaces. So those silicon surfaces are effectively our controls for the nanocones that don't have the coatings. And that's just because of the chemistry that we're doing. The silicon surface makes the most sense. There's a lot of very well-developed nanofabrication methods for silicon. So those are silicon dioxide nanoparticles. Very carefully, it would be my best answer. It took a lot of trial and error, and if you're interested in working with these, I can talk to you during the reception. But there was a lot of variables to modify in terms of the dry time and concentration and the angle of deposition, and it was a hassle to generate the monolayers consistently. And there's a lot of variables to discuss there.
Thank you, Evan. You talked about the history of using silver. Do you know much about like how long have been scientists and labs trying to make their nano spikiness surfaces?
Yeah, so other nanospike sort of formations have been used. I think a really great example, there's a Nature paper on using glancing angle deposition to form these sort of nano pillars. And some other ones that use basically they create like casts of the cicada wings and reproduce the columns using what's called nano imprint lithography. Most of them are relatively recent. And there's not work really yet using these kinds of structures to try to enhance the antimicrobial chemistries that we've been previously looking at. Oh, I'm so sorry.
Oils, that the thickness of the stone oils, like that kitchen microns. So how would you expect these to perform if they're kind of filled in with REU?
Yeah. So the question is, how would I expect these surfaces to perform if things like surface grime built up on the surface? That's a really great question. I don't know that it would be very genuine for me to sort of try to give you an exact estimation of what I think would happen. I think that it's likely that there would be reductions in effectiveness, especially if you have like a strictly antimicrobial physical structure, if it's coated. The great thing about like these nanosurface engineering kind of problems is that you can modify the wettability of surfaces, which has a major effect on the deposition of grime. And so a lot of other people designing similar nanosurfaces modify hydrophobicity to prevent the buildup of grime on the surface. And I think that could be a viable method if that was turning out to be a problem. Oh, awesome. Thank you, Dr. Graves.
If this is the mechanism to make these surfaces specific for any given bacterial surface species, I include the surfaces that are specifically found by a given.
That's a great question. I think that if you were targeting one specific species, that might be a viable method. For the specific engineering problem that we're looking at, trying to just generally prevent the buildup of bacteria on surfaces when we're looking at a variety of species across a variety of genera, it might be more effective to focus on more general solutions, but I think that could be a really interesting research direction.
We are welcoming up Mara Chin Goldberg, who is joining us from the University of Maryland, where she is beginning her junior year studying environmental science and math. This summer, she's been working with Dr. David Singleton in the Gunch Lab, trying to find ways to control fungi. She's going to tell us more about the fungal control in her presentation.
Hello, everyone. Thank you for being here. I'm going to be talking to you today about my project developing methods for testing antifungal bacterial inoculants on built environment surfaces. Yeah, so just for some background behind our research problem, we're concerned with mold in their residential built environment, which is a health hazard because it's sort of a persistent environmental exposure for residents. And so conventional cleaning agents such as bleach can get rid of the problem, but it often comes back due to just the conditions of the home. And also repeated use of bleach over time and other chemical cleaning agents can cause adverse health effects to the people living there and constantly using them. So for those reasons, biological agents are a promising alternative. So there are several existing commercial probiotic cleaners on the market, and they use bacillus strains, which are generally recognized as safe by the FDA. And so we use some bacillus strains in our experiments. And yeah, so we were looking at Aspergillus as a target organism for testing our antifungal bacteria. So Aspergillus is a fungal mold commonly found in flooded homes because it grows on built environment materials at a high relative humidity. And the specific species we're looking at was Aspergillus flavus, which is an opportunistic pathogen that also produces mycotoxins. So it can be a problem for immunocompromised residents because you can get aspergillosis from inhaling the spores, but also prolonged exposure to the mycotoxins can cause chronic health effects. And so the model Aspergillus strain we were testing was actually isolated from the PreMiEr home at Duke. Yeah, so we had 3 research goals going into the summer. Number 1, we were going to screen bacillus strains for antifungal behavior. Then we were going to develop methods for testing those strains. And then we were going to assess the ability of those strains to actually inhibit Aspergillus growth on simulated saturated drywall conditions. Yeah, so for the first part of my summer, I was looking at this library of bacillus strains and screening them for antifungal behavior. And I was doing this by setting up competition assays on agar plates, where, as you can see on the left, you spot the Aspergillus and the bacillus next to each other, and then let them grow over a week or so. And when they grow, one of 3 things can happen. Either the bacillus strain exhibits direct inhibition, which you can see on the top circle, where the Aspergillus colony is actually just not growing towards it at all, and that's because the bacillus is producing some metabolites that kill the Aspergillus. And some strains can also exhibit indirect inhibition, where they compete with the Aspergillus for nutrients and resources and space, and they just grow way faster. And so you can see on that second circle, the bacillus side has just completely taken over the Aspergillus, prevented it from growing any further. And then the third thing that can happen is that the bacillus has no inhibition or weak inhibition. And you can see that in the third circle, the Aspergillus has just completely grown over it. So we screened 14 isolates and also 3 strains from the commercial product. And we found 2 that were directly inhibitory and 2 that were indirectly inhibitory. So we continued with those 4 strains and also the commercial product mixture. Yeah, so then we moved on to playing around with methods for actually evaluating the efficacy of these inoculants. So first we tried using actual drywall, but there were many issues with this. It was difficult to inoculate evenly. It came with internal contamination. So yeah, using actual drywall did not work. So then we tried to just simulate the surface of drywall as if it was flooded by stormwater. So we set up an assay where we had 2 different nutrient conditions, either no nutrients or stormwater level nutrients, put craft paper, which is on the surface of drywall, on top of it, and then mix together Aspergillus spores and bacillus spores and inoculate that onto the surface of the craft paper. And so we incubated these for 4 days at high humidity, and then we sampled with petrifilm. So petrifilm is like desiccated agar that has some growth nutrients, but also limiting nutrients to prevent it from spreading very far, so that when you touch that to the surface of the craft paper, it picks up the fungal colonies and it turns blue where there was dense fungal growth. And so we developed a image analysis macro to quantify where the growth was the most active on the craft paper. So yeah, so for our initial experiments, we inoculated the Aspergillus alongside 10 to the 3rd bacillus spores, and so we didn't see any effects when we did this. On the left, the Aspergillus only control, that's the amount of growth that we saw in the craft paper when there was no bacillus. And so we were looking for any bacillus strain that would reduce that growth. And as you can see, none of them did that. So we repeated this with a higher concentration of bacillus spores. And again, there's no bacillus strain that decreased that growth. So then we also tried mixing together strains that had direct inhibition and indirect inhibition, and did it at the same concentration of bacillus as before. And as you can see, none of these had a very significant decrease. So at this point, we're thinking that maybe in these conditions, just Aspergillus is way favored and grows much faster. And maybe this competition might be preventing the bacillus from producing the metabolites it would be using to directly inhibit the Aspergillus growth. So we wanted to see if the bacillus would be effective if they were able to get there first. Yeah, so we did some modifications to the previous method where we inoculated and incubated the bacillus first, and we also added a condition with high nutrient media to amplify any possible effects of the bacillus. So yeah, as you can see, there was a visible effect of adding the bacillus first. So on the left-hand side of all of these pictures is the high nutrient condition, and you can see that where there was only Aspergillus and no bacillus, there's a lot of visible growth. But where there were the directly inhibitory strains, especially WS1, there is no visible growth. And the strains that were indirectly inhibitory, remember the ones that can grow faster but not necessarily produce the metabolites that killed the Aspergillus. On those surfaces, we saw that they sort of pushed around the Aspergillus, but weren't as effective at reducing the growth. And so, yeah, so these are some of the petrofilm images. On the high nutrient condition, we could see that strains WS1 and B6, which were the directly inhibitory ones, inhibited nearly all fungal growth, and using the commercial product reduced fungal growth. And on the low nutrient condition, the results were not as dramatic, but they were still noticeable, as you can see by the side-by-side petrofilm plates, and strains WS1, B6, and also the commercial product reduced fungal growth. So yeah, this is the results for the low nutrient condition, which is simulating stormwater saturation. And as you can see, the directly inhibitory strains were effective, with strain B6 being about as effective as the commercial product. And one of the indirectly inhibitory strains was also effective. So yeah, our overall conclusions, it seems like inoculating Bacillus endospores at the same time as Aspergillus has no effect on the Aspergillus's growth, but if you allow the bacillus to germinate overnight, it does reduce growth. And furthermore, in terms of method development, petrofilm is a promising method for assessing inhibition of fungal mold growth on surfaces, especially moving forward towards testing in places like the Duke home, the PreMiEr home, because you can use it on any surface. It's easy to store, and it also captures and preserves spatial information. And yeah, for further use of this, the image analysis might need some optimization, but yeah. And further directions of this research can focus on more development of using bacillus as a preventative measure, such as in what conditions does it work the best, how long would it work for, and also just some characterization of bacillus growth from endospore form on simulated saturated drywall surfaces, and as well as how long it would take to produce those direct metabolites and under what conditions. Yeah, huge thank you to everyone I worked with, especially David Mentor and Philip, the RET, that helped with a lot of the work, and the REU cohort. Yeah, are there any questions? Yep. Okay.
Based on the results that you're seeing, you feel like pre-application being more effective in the longer scheme of things where we're using bacillus as a preventative control. Would it have to be used as a preventative control, grow bacillus on your walls in anticipation of potentially getting mold?
So the question was, will bacillus have to be used as a preventative control as opposed to a remediative? Yeah, sort of yes. It seems like it would not be effective if you just applied the spores when Aspergillus was already growing. It seems like that would not be as effective. So yeah, moving forward, looking at bacillus as more of a preventative approach, I think is the way to go. Thank you for, yes, what's up?
Preventative material when it's manufactured, or would it be sprayed under before you see any mold growth? And then it would, I guess I'm also curious about you don't also want the bacillus to be growing on the, even though it's not hurt anybody, I guess.
Yeah, it's kind of sticky. So.
I'm curious about
Yeah, thank you for the question. The question was if we envision the bacillus sort of being embedded in the material or like sprayed on the wall. So there have been studies done where people embed bacillus in like paint, and that has been shown to have an anti-mold effect. For more of a product that you could apply at will, I could see there being a sort of spray that you spray on your wall, sort of if you know that there's going to be like a big storm coming through, maybe you would put it on your the bottom of your basement walls just in case there's flooding. But I do agree that having a lot of bacillus growing on your wall might not be desirable for many people. Thank you for the question. Yes? Yeah. So the question was if I knew to what extent the direct metabolite inhibition versus the indirect inhibition was affecting the fungal growth. And I think that at least on the craft paper surface, it seems like the direct inhibition has a stronger effect. This is when it produces metabolites that directly kill the cell walls of the fungi. And I think this because when we tested it with the indirect inhibition, it's still like the aspergillus mostly still grew, even though it might have been like moved around. It seems like it still found those pockets to grow in. Whereas when we test it with the bacillus that actually are producing the metabolites that it cannot tolerate, then it just doesn't grow. Yeah, that's a great question. The question was, can you characterize the metabolites and use them as their own remediation? There's been a lot of work done, specifically the strains that we use are Velazensis, and those metabolites of related Velazensis strains have been characterized to be like surfactants. And so that would just sort of be a similar chemical cleaner that people could use in a more traditional method if we were to use that as its own targeted remediation. Are there any more questions? Yeah.
So it's important if you are going side by side at the same time, both the bacillus also create antifungal compounds. Why wouldn't it inhibit this?
Yeah, so the question was why we think the bacillus might not be inhibiting the fungi under these circumstances. I think that it might be because they are not like the aspergillus are taking up some of the nutrients, and the bacillus need to have a certain threshold of nutrients to produce the metabolites because they need enough carbon to actually like build the surfactant like compounds. It could also be so the Velazensis sort of like quorum sensing pathway has not been super well characterized, but some related strains, like I think that some other bacillus strains, their quorum sensing pathways have been linked to genes that produce surfactants. So it could be that when they're there with the aspergillus, they're not in high enough concentrations to start producing those metabolites, which is why they might start making them when they are able to colonize first and grow. But that's just, that hasn't been tested. Anything else? Thank you.
Okay, keep Bacillus velazensis on your mind as we introduce our next speaker, Niobe Fleming, who's joining us from Nova Southeastern. Niobe is studying medical humanities and biology and has been working with our groups here at UNC across the Brown, Turpin, Noble labs. Yeah, okay, Niobe.
Hi, my name is Niobe, and I'm going to be talking about characterizing bacillus growth, aerosolization, viability, and distribution in built environments. And hopefully I'll answer some of the questions you guys had for Mara. So we are evaluating Bacillus velazensis as a model indoor biotic inoculant. So what is a biotic inoculant? So these are preparations of living microorganisms that can be intentionally engineered to provide a benefit. In this case, ours is fighting fungal molds in homes. So to do this, we have like three main prongs. So the first thing is characterizing the growth of bacillus. So we want to see, can we prepare this as a biotic inoculant? How does it grow? How long does it take to grow? And then we want to see how long does it survive when we deliver it via humidifier. And then we're going to apply this information to the Duke test house to see how this could look in other people's homes. So the first experiment we took a look at was determining when is it in exponential growth, when is it doubling. So to do this, we had a growth curve experiment, which I'm going to talk about very shortly. And then we wanted to see, is there a difference between the vegetative cells and the spores when we use them in an ultrasonic humidifier. So that's our mode of delivering this bacillus. So we like to aerosolize it. And then we want to see, where does this aerosolized B. valencius deposit? How far does it travel, and which concentrations affect that? So the first thing we did was establish a growth curve. So we started by growing an overnight culture, which was incubated at 28 degrees Celsius and shaked at 200 rotations per minute. So then we pulled from the overnight into 3 replicate flasks, and we allowed those to grow over the course of 3-4 hours. So from hours 0 to 12, 0 to 6, we sampled once every hour. And when we sample, we're putting some onto TSA plates and incubating those to allow them to grow to see the colony forming units. And then we're also taking the optical density using a spectrometer. And then from hours 6 to 12, we're taking every 2 hours. And then we take again at T24 in the morning, which is with the hope of constructing a growth curve. So the growth curve, for those who are not familiar, we're supposed to see this lag phase here where we're not really seeing significant growth. And then we see this log phase, this doubling time, which is what we're essentially interested in, because when we do our humidifier viability experiments, we want the bacteria to be in its doubling log phase, because that's when it's going to be the most viable for growth. And when we're looking at the humidifier and we're trying to measure how does this grow and when, we want to make sure that we are giving the bacteria essentially its best shot at growing and that there aren't other factors. And then we're going to see this stationary phase where there's no growth, and then this plateau, which we won't see because this is about after 48 hours or so. So let's take a look here. So we have our optical density and we have our colony forming unit. So I'm going to direct your attention here to the colony forming units graph. And this is the time point in minutes. And then this is the log 10 of the colony forming units. So you can see what I was talking about, this lag phase in the first couple time points where we're not seeing growth. And then we kind of see this exponential growth, and there is a little dip here. But this is essentially our log phase. And the bacteria essentially reaches this around 6 hours. And then we see this kind of plateau out. And we see something similar with our optical density values where we have this kind of log, and then we grow, and then we plateau out. And around 12 hours is when it's hitting this like stationary phase. So the next thing we want to determine is, does ultrasonic humidifier operation affect the cellular viability? So to do this, we have our humidifier and we have our control. So we create a solution of 1 liter of PBS between, and we add 250 milliliters of our culture. It's the same protocol for both the spores and the vegetative cells, and those are grown into LB and then added. For our liter, 1 liter goes into the humidifier, and the humidifier is turned on and ran for 7 hours. The control is just in a bottle sitting in the biosafety cabinet. From there, very similar to our growth curve, we sample incrementally once every hour for the first 3 hours. And then from hours 3 to 7, we take every half an hour. And from there, we wait to get those colony forming units. So we did not take the optical density from this experiment, but that is something we considered doing for our next steps. So here's what we found, and it's actually quite interesting. So we'll start with the vegetative cells. And I do want to start by saying why we decided to look at both the vegetative and the spores. Most commercialized products that are using bacillus are in the spore form, but we also kind of wanted to take a look at what it looks like vegetative to see that. So the control and the humidifier kind of track each other towards the beginning, and then we see this drop off around after 5.5 hours or so, where there isn't any growth. We're not counting any colony forming units. And our x-axis here is time, and then our log colony forming units. And then for viability within the spore form, they continue to kind of track each other. We don't really see as much of a difference or drop off in our spores like we do see for the vegetative cells. So for our next steps, we wanted to see how can we translate this to the Duke test house. So this is a model of one of the rooms that we've been working with in the test house. And these are 1 meter squares, and this kind of smaller square here is 4 by 4 inches. So what we do is before we turn on our humidifier with our bacillus solution in, we place settle plates, and we allow those to settle for about 90 minutes. And then we also swab the center to kind of see what is it looking like before we introduce bacillus to the area. Then we turn our humidifier on. It's placed right here in the middle. This is centralized in the room for 90 minutes, and we allow that to run. We take the swab again, and we also use those settle plates to see how much bacteria is going to be on these plates and where will it travel. We also do wall sampling as well. So this is kind of the layout on the wall. This is actually pretty accurate if you've been to the house and how it looks. And we swab this inside here. The AC systems are turned off and the windows/doors are closed, but there could be some variability with the HVAC system because sometimes it doesn't truly turn off. So here's some preliminary data we have here. This is just some of our early trials. We are still developing the methods for collection within the Duke test house. So square HF, I just want to remind you, is where our humidifier is placed. And it's interesting, right next to it, IF is where we have our highest recovery, but on the other side, it's not nearly as high. So we have hypothesized that there could be some type of lean with the humidifier itself, or there could be airflow, other factors affecting why there's so much recovery here. It is not unexpected that the majority of recovery that we see is right near where the humidifier was. So some of our takeaways could be that perhaps we would use multiple humidifiers, perhaps we would increase the concentration or play around with some of the other variables in the house. So in conclusion, we have to kind of grow the bacteria first, and then we wanted to see how it grows in the humidifier, also that we can get to this application of how this would look in the Duke test house and hopefully how it would look in your house. Thank you to Dr. Megan Lott and my mentor, M. Norton, and the Brown Lab, and of course, the PreMiEr REU cohort. Any questions? Yes. Good question. Yes, so the question was in the heat map diagram where the door is in the room. So this is the floor, the door would be right here around LF, OF. I'll divert your attention back here. You can see kind of where the door is in relation to squares LF, OF, and IF right here on the side.
So do you think they made that well from maybe that?
Yeah, that's definitely something we've hypothesized. We do have the doors closed, but the HVAC system can be kind of hard to totally shut off. So there could definitely be some of that air current stuff happening that's moving that bacteria around. This picture here is actually pretty accurate to how our humidifiers work. So they do go straight up. The nozzle has a bit of a lift, but that could definitely be some of that air current coming in and moving it around. Yes. Yes. So the question was looking at the differences between the vegetative and the spore-forming graph. Are they killing at the same rate before the drop-off, and what are those differences really showing? So I think that's a great question. So if we look at the colony-forming units here, from the start, the highest we typically reach is between this like 5.5, and then we go up to 6. But when we look at the spore, even before we have this drop-off situation happening here, they typically stay between 750 and 8. So they're typically higher up. So we do record more colony forming units when we pull from the humidifier within the spore form, which kind of leads us to think that these spores are more resilient when put into the humidifier. The ultrasonic humidifier does add some heat and there is movement within that. And it's not conditions that are ideal for the growth of this specific bacteria. So we are thinking they're more resilient in the spore form, which would also match with the literature and a lot of products using specifically the spore form. Not to say that there isn't viability within the vegetative state, because we do see here for the first few hours that there is some viability here. And a lot of what this data was used for was when we're in the Duke house, or if this becomes a product that is commercialized, how long would we say to run this? How long would we want to have this turned on? And when we do our experiments, do we need to be in there for 6, 7 hours when, if we're using the vegetative state after hours 5.5, there's nothing happening? Thank you. Yes. Yeah. So the question was, did I see any difference in deposition between the spore forming and the vegetative task? So for the most part, I would say they were really modeling each other up until around hours 4 to 5. They were pretty similar. There is some like marginal difference with the colony forming units task, but they were very similar, especially when we looked at the plates. There was like key similarities within the colony morphology. When we look at this strain, they typically tend to be smaller and circular, and we saw them looking pretty similar when we were looking at the plates and analyzing the data. I think our main difference is the time. Thank you. Any other questions? Awesome.
Hello, it's Rachel. Can you hear me?
Yes. Yes.
Yeah, great presentations from all so far. I've just had an absolute blast listening and some really, really great work. I have one practical question and one kind of more microbiology question. So I know a lot of the industry partners that build these microbial products, they have an activator in their product that allows the bacillus basically to remain stable when it's sitting on the shelf. And then whenever it's sprayed, it's actually activated and begins that growth. Can you just kind of speculate a little bit on how that kind of the idea of some sort of activating agent could be incorporated into this, either a humidifier or a diffuser kind of a scenario?
Thank you. Absolutely. So when we go back here to kind of establishing the methods, or I'll actually look here, that is something we were concerned about because there is such a time difference when it comes to viability. So if this were to become a product, something we talked about was, would this be shelf stable? How long can you have this for? Are there conditions this needs to be stored at? Does it need to be refrigerated? So we did play around a little bit with that viability. So when we make the for this, we do start with an overnight culture. So when we make that overnight culture, sometimes it's made the night before. We did it one time when it was made a week in advance. And we did see differences when we used the overnight that was about a week old versus the one that was newer. And kind of playing around with that time, we do see a difference. So I think that's something we've thought about, that an activator would totally be necessary. We would like this to remain viable for as long as possible when it's used. And it's also going to be put into the humidifier, which is a different stressor on top of already not being in its ideal conditions. So I think that an activator would be very necessary. I haven't really heard too much about with the spray, but I think that could be productive as well, especially because these bacteria are going to be aerosolized, and that's a similar method.
Great. The specific question that I had really kind of almost combines the work for you, Niobe and Mara, was, is there any chance that tracking toxin production is as valuable as tracking cell growth? So it strikes me that even though the bacillus in certain circumstances might not be having an impact on the actual aspergillus, that maybe there's a reduction in toxin production that actually is valuable. So I wondered if between the two of you, if you could kind of talk about, how you might track toxin production. Like if you were here for another year and had unlimited resources, either one of you, like how you could investigate that, or based upon what you've learned, that kind of a next step in terms of, because maybe we don't have to get rid of all mold, which we know is impossible, but somehow maybe reducing toxin production is really the valuable step of the bacillus treatment. I wondered if you could just kind of speculate on that a little bit.
Yeah, Mara, do you?
Okay, hello. Yeah, thank you so much for the question. That definitely would be an interesting thing to track. I think that, yeah, it's not something that we had considered before because I think that this species of Aspergillus is known to produce these mycotoxins, but it would be an interesting experiment to track the production of mycotoxins and if that decreases in the presence of the metabolites that directly inhibit it, or like even if the metabolites aren't in a high enough concentration to kill it. That would definitely be a cool thing to look at in the future.
Yeah, I would agree with Mara. Piggybacking off of that, I think it could also be interesting to test kind of the methods of delivery, of how we could deliver this bacillus, for example, like aerosolized bacillus, seeing how that could impact the growth of Aspergillus, for example, on a plate, similar to how we do in the Duke house. A lot of delivery with bacillus, like onto fungals, is sometimes it's spread plating, it's streak plating. So I would also want to add to what Mara said, to play around with seeing how the aerosolized bacillus itself can fight Aspergillus and other types of fungi.
Great. Thank you so much.
Thank you.
You all did great.
Thank you.
We've got our final presentation, last but not least, Mr. Will Medley is joining us from Elizabethtown College, and he is studying mechanical and electrical engineering, but this summer he's specifically been studying volatile organic compounds. So, Will.
Oh yeah. So today I'll be presenting my presentation on real-time VOC sensing for fungal detection in indoor materials. My mentors were Dr. Clara Eichler, Dr. Megan Lott, and M. Nortman. So first, why this matters. You probably think of fungi, you think of physical fungi growing on walls, but if fungi is growing on walls, it's likely been growing for days, if not weeks, which can be harmful to people who are immunocompromised or people who have respiratory issues. So we wanted to figure out if there was a way to track VOCs, which are volatile organic compounds commonly emitted by fungi, early on before they are visible. A problem that we had to address was that fungal VOCs often overlap with material VOCs, and a way that we were trying, like a way to identify and trying to differentiate between material VOCs and fungal VOCs was to not just trust one sensor alone, but to look for multi-sensor fingerprints. So the hypothesis we came up with was real-time VOC fingerprints can separate major source classes, but early-time fungal growth on real materials will be harder than fungi grown on PDA. And the three questions that we were answering were, do fungi controls in indoor materials produce different multi-sensor VOC fingerprints? Can early penicillium growth on drywall be separated from wet drywall alone? And can machine learning differentiate between selected VOC sources? The figure you're seeing on the right is an example of what a fingerprint is going to look like. It is a normalized heat map, normalized by row, and the y-axis is going to be the material or the fungi. The x-axis is going to be the sensor. So we're using the Sensory, which is developed out of Guiyan Chen's lab. It's an 11 sensor array, including a VOC sensor, a formaldehyde sensor, ethanol sensor, methane sensor, and 7 other sensor channels. And for the figure, you're looking left to right to see that they are individual between the material. Just to preface. The setup was a 10-liter dynamic flux chamber pumped with a constant flow rate from ultra zero grade air through a humidification system for 15% to 90% relative humidity to replicate common household relative humidity and then also fungal growth, like ambient conditions for fungal growth. The sensor is attached to the top of the lid of the chamber. And we also collected thermal desorption tubes in duplicate for GCMS analysis later on. And we also used, again, real-time sensing to actually have a figure to see as we put things in the chamber. The sources tested were common household materials such as MDF, plywood, painted materials, caulk, air fresheners, and then the fungi that we tested are pictured there. So for our workflow, it was talked about previously, but we looked at a delta from baseline. So we blanked the chamber for an hour and a half before we put the sample in, and then we cut that blank for until 2 minutes before the sample was put in. So that way all of the figures were plotted from the baseline of where the chamber was blanked, not from zero. So we could have accurate like changes. We cleaned the visualizations by using smoothing and clipping methods. For smoothing, we looked at the 10 time points, it took the median to get rid of any extreme outliers or any extreme spikes from the sensors. And then for clipping, we looked from the first to 99th percentile to get rid of any extreme outliers as well. And these were fed, the visualizations and the CSV files were fed into a repeatability, similarity, and machine learning algorithm. We used 3 different machine learning models, which was extra trees, random forest, and gradient boosting, which I can talk about more later, but I don't have time to now. And the key comparison metrics were a Pearson correlation, a Spearman correlation, cosine similarity, and mean absolute difference. So result 1, again, the figure is a normalized fingerprint for the rows individually for what the materials were. The top 5 common household materials to the fungi had above a 0.85 Spearman correlation, meaning they were highly similar. And you can see with some of the fingerprints, they look very similar to the fungi themselves. So a real indoor sample may not be easy to classify by fingerprint or shape alone because material background can mimic biological signal. For result 2, early drywall growth was not clearly separable. The penicillium inoculated drywall was highly similar across all the testings that we did. We did a 7-day, 12-day, 14-day, and a 19-day with a Spearman correlation of 1 for the 7-day, meaning they were the exact same. Spearman correlation of 0.964 on the 12-day, and then at 0.991 on the 20-day, with a mean absolute difference of I believe it was the 19th or the 20th day sample of 0.08, meaning they were still highly similar. I do want to say the 12-day Spearman correlation did pique my interest only because we tested all these materials previously that were just like on PDA plates, and the penicillium toxicarium was differentiable from PDA by the CH4 signal, which was highlighted again here on the 12-day sample. So I thought like maybe it was changing, and it could be like metabolizing at that point or like just emitting from VOCs at that time, but it did go back to being not differentiable on the 20-day sample. And then result 3 for the machine learning output, this is a confusion matrix with the goal being you want to see the dark blue squares on the main diagonal, with the x-axis being the predicted class, the machine learning predicted class, and the y-axis being the true class. So this had a 40% accuracy, predicting 36.5% of the fungal growth on PDA correct, and 83% of the household materials correct, which sounds good in practice, like looking at it from that, but it's not great. And then when you kind of blow it up to incorporate the top 5 similar household materials and then trying to get it class specific, you can see the B.AD fungi was classified as Xylella cubensis almost every single time. And then you can see that the penicillium toxicarium was never classified as penicillium toxicarium. So this had an overall accuracy of 19.8%. So in conclusion, the sensory does show a promise for screening indoor fungal VOCs. It's just in a noisy environment, it is really hard to differentiate between household materials and fungi. The VOC patterns do overlap with a lot of common building materials, and then machine learning can distinguish fungi from PDA controls, but it struggles with household materials. For future work, we want to increase biological replication. A lot of these were tested on only 2 or 3 trials, and they use window analysis for machine learning rather than trial analysis. And ideally, you want to use trial analysis to have the entire trial be taught and learned on. We also want to expand the library for household materials and for fungi, just trying to have a clear fingerprint for what's being sensed in a noisy environment, and then validating all the sensor responses with GCMS, and then improving multi-class machine learning models for real indoor environments. I do want to thank my mentors and the cohort. It's been a great summer, and I love my time here. Are there any questions? Yes.
I know your focus was in machine learning, but I was wondering what were the material or their high speed stage or were there already like were there any visible signs of things probably that you can say maybe the contract?
So the question, are you asking specifically for the drywall? Okay. So the question was asking, was there any physical signs that the inoculated drywall was growing fungi? Okay. The answer was no. We couldn't see any physical fungi being grown on the drywall yet. We do have pictures of all the time points, and it looks identical in each one. So there is a possibility that the inoculated drywall is not growing fungi, or I suspect that the wet drywall gives off a stronger signal than the fungi do themselves. So it's just masking the signal of the fungi as well. But yeah.
Back a little bit to
Mara and Dr. Singleton's work with the drywall, it seems like possibly uninoculated drywall already has a pretty diverse and intense sort of fungal community going on. Do you think that previously present fungal signatures might be masking the introduced fungal signatures in the drywall experiments?
The question was, do I think that, or from looking at Mara's presentation, that drywall already has some fungi signatures in there, do I think that that could be masking the inoculated fungi signals? My answer is no, only because we have a background for drywall by itself and wet drywall, and both of those were, like for machine learning, it could differentiate between those very well. I don't think that if there was any fungi in the drywall, that it would be masking the signal of the inoculated drywall. Or if the signal would be masked, I think the signal would be masked more by the wetness of the drywall than anything, if that answers your question. Okay.
You have that question.
Yeah.
And it's talking about the combination. So, the question is, if there are chance that combinations. Like
Oh, okay. When we were testing the signals, we used a sensor array. It was 11 sensor channel. And the fingerprints were used in the machine learning to differentiate between what was like the materials. It wasn't just like one sensor channel at a time, if that answers the question.
Yes, and then, well, could you further explain?
Oh, sorry. Yes, so for the time series, we pulled the min, max, median, mean, and then the slope of the curve for how quick the rate of change was from the baseline. And then that was simplified into just a point figure. And then we use correlation methods for how similar that was to other signals. We fed everything in, and we used the raw data, so we didn't use any clipping or smoothing for the machine learning.
And then,
one more time, so sorry.
I might ask her. Wait, do you ask her?
Oh, yeah, can you say your question out loud, please?
Sorry, there's a bit of a delay and I'm having a hard time hearing you all. What I was referring to is kind of like you already said in your kind of next steps replication. So what I was thinking about was kind of replication of the measurements, combined sets of measurements over time, like basically combining events to create a bit more of a combination of signals that could be more predictive.
Yes, the trials are averaged between like different trial runs. It's like for MDF, we did a total of 3 trials, and those trials are averaged for the machine learning. So like it simplifies it into one like MDF signal.
What was the length of those trials?
It ran for an hour and a half.
Okay.
Okay.
Great. Thank you.
I'm so sorry. Can you say it one more time?
So I just yesterday we talked about combining the data from the set of.
Oh, okay. I have not done that yet. Sorry.
Okay, we'll follow up.
Okay.
No, thank you so much.
Y'all, that brings us to the end of our slide deck, and I think to the end of our presentations. Thank y'all so much for joining us this morning. It's been great to hear about this research. One last round of applause for our REU and RET participants. A couple of quick housekeeping items. If you parked and you need parking validation, please see me and make sure that I've been able to get your license plate over to our business office for parking validation. For folks in person, we're gonna head outside and I'm hoping Med Deli is waiting. Waiting for us. Folks online, thanks so much for joining. Folks in person, please continue these conversations about research. Please ask the REUs who is in this clean suit in this picture and how they spent their off time. So, thank y'all.
Thank you.